Hi, everyone. I'm Seda Kurt from the International Biogeography Society, and welcome to the first Funk lecture of the year. Today, we're hosting Maria Dornelas, and she'll be presenting about biodiversity change. And while we're waiting for people to drop in, I would like to talk about upcoming events. The next speaker of the Funk lecture will be Jose Alexandre, and the talk title is Divide and Conquer Biological Diversity as a Source of Underdetermination in Macroecology and Biogeography. And also the next biennial conference is in Prague, and the society is working on it a lot on these days to offer you an excellent conference as always. And we will also announce the next workshop soon. And after Maria's talk, we will have 20 minutes Q&A session, and you can raise your hand to ask your question or type in chat during the talk or after the talk. So let's wait a little bit more, and then David will introduce Maria and start the session.
Everyone, welcome to today's seminar. Good evening for you in the Eastern Hemisphere, and maybe good morning, good afternoon for each of you in the Western Hemisphere. And it's a great honor to have here today to Professor Maria Dornelas. Maria took a Bachelor of Science in the University of Lisbon, and she moved to Australia, University, to do a PhD thesis. So it was mainly focused on studying biodiversity patterns in the light of neutral theory. So the research of Professor Maria Dornelas basically navigates across the disciplines of macroecology, community ecology, and biogeography. She's a really good example of a scientist that is able to link at a really high level theories, big data, but also fieldwork and experiments in the field. Maria just come today or yesterday from the Red Sea, from doing some fieldwork and teaching on the side. So the main systems of Maria are mainly marine and aquatic systems, with a special emphasis on coral reefs. But at the same time, Maria has been one of the leading forces of what is maybe the largest database on abundance over time, that is called BioTIME. And so I think it's a great honor to have here today Maria Dornelas, and given the wide and broad impact of her work. So Maria, welcome. Thanks for being here with us today, and the stage is yours.
Thank you very much for the invitation to give this talk. If you bear with me a second, I will share my slides. And I hope you can see them. Please tell me if not. Okay, so as David mentioned, I'm only just back from Egypt, spending 2 weeks underwater. So forgive me if I start this talk with a bit of a reflection, because I guess spending 2 weeks on the water, watching students learn how to collect data while diving, afforded me, I guess, the removal of distractions, and the fact that I wasn't actively collecting data myself led me to spend some time meditating and reflecting on the nature of data in science, and also, I guess, the differences between data and knowledge, because I guess it's really insightful to watch how surprised students are about the pervasiveness of error in the data that they collect, because error is everywhere. And at some point I found myself thinking about Plato's allegory, and thinking about how data are just mere shadows of reality, and we're kind of stuck in a cave trying to make sense of them, of the data, but also structured observations help us, are really precious in helping us make sense of the world, and in a way data and our reason is all we have, yeah, and I guess this talk is about using data, using data from the past to look back on how we think biodiversity has changed in the recent past. So I've structured this talk into 3 different sections. The first one, and I guess I'm going to be talking about some recent work, as well as some work that's still kind of work in progress that we've been doing and trying to make sense of how biodiversity has changed in the recent past. And I've structured the talk into 3 sections. The first one is focused on the data that we have been assembling and using, which consists of biodiversity time series. The second section is focused on the patterns that we have uncovered in these data. And the third one has some reflections on what I think we can and cannot say with what we have uncovered so far. Okay, so the data is not mine. It's the collective effort of over 300 scientists from across the world. I have been privileged to be involved in collating and assembling this data, and I'm referring to the BioTIME database, which is a database of a biodiversity time series for the Anthropocene. We made a database public in 2018, and it was a bit of an odd manuscript in that we had 2,000 words of text, but we had 3,000 words with the author names and their affiliations, and 1,500 words of acknowledgement. So a very lopsided paper, but rightly so, because collecting data is expensive in time as well as research funds. And so it's only right that the people who collect the data get credit for their work, as well as responsibility for any error that the data may have. So at the time when we published this data paper, the database had not quite, but approaching 9 million records, 8.7 million records of where a record is the observation of the abundance of the species within an assemblage at a place and a time. And at that time, we had observations in over 500,000 places or locations here. And that database included around 40,000 species. And these, although these data came from 361 different studies, where we define a study as a methodology of observing an assemblage. So it could be anything from bird transects to fish trawls to all kinds of things. We didn't discriminate on locations or taxa within BioTIME. And because we're talking about a time series, I should also mention that the data spans from the late 1800s to the present, although really the overwhelming majority from the past 50 to 40 years. So I'm really proud of BioTIME. Feel super privileged to be part of the team that leads it. There's a core team in addition to the hundreds of data contributors, which includes Anne Maguren, who led the ERC project that started this, myself, Amanda Bates, Laura Anton, Frey Moyes, and more recently also Viviana Brambilla and Cher Chow. And we're always doing improvements to or trying to improve the database. This little video that I just played is a Shiny app that Cher Chow put together to help us or to facilitate exploring what data we have in the database. So like I said, I'm very proud of this, but also I am very keenly aware that it's far from perfect, that we have lots of blind spots. I guess I would argue more blind spots than spots where we actually have data from. And this really matters because it means that we don't know what's going on and including we don't know what's going on in some parts of the planet where we think that drivers of biodiversity change are particularly high. And I say this, so this is a map that comes out, a map of cumulative drivers of change. It came out of an ST working group that I co-led with Maria Connor and Sarah Sub, the S-Change working group. And it's a paper that's led by Diana Bowler, Leticia Navarro, and Amanda Bates. And in this paper, we combined marine and terrestrial spatial layers of putative drivers of biodiversity change, and then we asked the question of how do they, how do they accumulate? The analysis revealed that are different combinations of drivers of change, what we call in the paper anthropogenic threat complexes. And it also showed that there is a great deal of spatial autocorrelation among most of the drivers. Largely, we have more drivers of biodiversity change in places where we have more humans, which makes sense. The one big exception being climate change, which is largely uncorrelated with the other ones. So, so we know that there is this heterogeneous distribution in space of the of drivers of biodiversity change, and we know we have special gaps in our biodiversity observations. One thing we did ask is how do the two things combine? And this is work that was led by Gergana Daskalova. And what Gergana did was sample the planet randomly. That's what the gray dots in these plots are. And then created a multivariate space of global change drivers. And what she did next was to superimpose the locations of biodiversity time series onto this multivariate space to figure out what parts of this of global change space are we sampling well, and where are we blind? What are the things that we have not really observations as to how things are changing? And the results are, I guess, somewhat mixed. There's definitely parts of this gradients in biodiversity or in driver intensity that are very poorly sampled. Somewhat worryingly, the biases are pretty much common across the different large databases of biodiversity. So in this case, Gergana looked at not just BioTIME, but also the Living Planet database and the Predicts database. Well, for BioTIME in particular, but also for the Living Planet database, the marine realm is somewhat better covered than the terrestrial realm. Okay. So this is somewhat worrying, though it tells us that we have important gaps in our knowledge, and we are working in trying to make this problem smaller. We're actually currently working towards the release of the next version of BioTIME. Again, still a work in progress. But we have so far grown the database substantially. We now have over 13 million records from 700,000 locations, and we've nearly doubled the number of studies. 605 is the current tally. And also, very, very importantly, we invested significantly in terms of cleaning up the data more, and specifically, we invested significantly in terms of cleaning up the taxonomic names of the data that we have in BioTIME, while recognizing that this will always be a work in progress. I mean, among other things, because taxonomy itself is not, we're not finished with that, are we? So it's like it's always changing anyway. So look out for this, and also if you want to contribute data within the next month or so, we might still have time to include it in version 2.0. Another exciting development with regards to data is the something called a new database called BioTIME, and this is being led by Janset Smith, Marina Rillo, Charlie Hall, and Seth Finnegan. I'm a very minor actor in this, but I wanted to highlight it because I'm pretty excited about it, and what this great group of people are leading is linking up modern data with fossil data, and specifically with subsetted BioTIME data that meets certain criteria, and we're combining that with lots of different fossil databases and data sources, with the goal being expanding substantially to millions of years instead of a couple of decades, the temporal horizons that are covered in these biodiversity databases. So this again is going to be a data paper that is very, very nearing submission. Okay, so I think those were the main points that I wanted to talk about the data, and I hope it's clear that it's not perfect, but there's a lot of it, so we should be able to learn some things. And so the next section of my talk is focused on the patterns that we have found so far by looking at these data. And I'm going to start with the first paper that we published that was used the BioTIME database. It has a few years now. It came out in 2014. At the time, the database was way smaller. We had 100 different studies in it, and what we did in this paper is fit a hierarchical linear model across these data to estimate trends in species richness, which we have on the x-axis here, sorry, and the y-axis here, through time, which is the x-axis, across the 100 studies that comprise the database at the time. And what we found is that contrary to what we expected, we saw a variety of trends, both gains and losses of species richness in the different studies that are the different colored lines we see here, which balance each other out, so that if we estimate a slope that is common across all the studies, we end up with a slope estimate that is indistinguishable from zero, which is the black line here. So this was somewhat surprising for us. We also looked at compositional change using the Jacquard index, which goes from 0 when two samples don't have any species in common to 1 when the same species is present in the two, like when all the same species are present in both samples. And we looked at how this metric changes with time from the first year of the time series to each subsequent year. And what we found was a pretty systematic decrease in similarity, which tells us that composition is changing through time pretty consistently. Now, out of the 202 lines that I'm showing you here, one was in sharp contrast with what we think we know about biodiversity change, for example, in terms of what we know about extinction rates being elevated, and etc. What we've been doing in the past few years is to try and reconcile these things by largely by dissecting the variation in these lines. So instead of focusing too much on the average line, what we've been doing instead is looking at what could be driving all of this variety of trends. The next analysis that I want to mention focus specifically on geographic patterns in the variation of biodiversity change. We kind of ask the question, where is the species richness changing? For this analysis, we tripled the amount of data that we used. One very important improvement that we also did at this point was the standardized spatial scale. We also used different types of models, and in this case we used Bayesian models. But despite all of this, we ended up in a not too different place from the point of view of what the average slope estimate was for the planet, in that it was still indistinguishable from zero. That's what this black dot is here, with the confidence intervals around it. But still, there is a lot of variation, and so we wanted to focus more on the variation. And so we color-coded the marine biomes of the world for where we have data, according to whether their average changes, species richness was above or below the global mean, the above being in blue and below in the red and the warmer colors. And what I kind of see in this map is that most of the red is towards the tropics, and whereas most of the temperate and polar regions are colored in blue, which would be consistent with polewards, I guess, potentially shifting and the latitudinal gradients of diversity. Now, in the terrestrial realm, patterns are far, far messier, and I'm not sure I can make sense of them, but also remember that the terrestrial realm is what BioTIME is far more biased, and so to some extent we should take this with a greater pinch of salt. There have been times where I've shown this map and people have said that if you squint, you maybe see some evidence of more red, where we have higher concentrations of human population. I'm not sure I see that, if I'm honest. Again, huge pinch of salt. What we see again is very, very prevalent, very high prevalence of high turnover. The estimate for the planet is still a great deal of turnover. That's what we see here in the black dot and with the confidence intervals around it. There are also some interesting patterns in terms of the spatial distribution of turnover, but they're more interesting if we actually compare the marine and terrestrial realm, in that we see some evidence that turnover appears to be faster in the marine realm than the terrestrial realm. So we've done a similar thing here. In blue, we have things that have changed faster than the global average, and in red, we've covered the things that changed more slowly than the global average. And we see that most of the marine realm is blue and most of the terrestrial realm is red. Okay. So I should have said, and I forgot that this geographic analysis, it also came out of the S-Change working group and was led by Shane Blows and Sarah Supp. And so far, I have largely been talking about changing biodiversity at the local scale, but looking at the broad brush across the entire planet. But for a second, I would like us to just focus, sort of zoom in on one example, because there's a very important point that I would like to make. And because I did my PhD in Australia, I still go back every year. I think I want to illustrate it with an example from there. And so I'd like to take you to compare two places, so Lizard Island and Sydney. And the point that I want to make is that we see a great deal of change, and one of the things that we see sometimes is regarding coral reefs and the polewards and extensions of coral reef species. And here is a photograph of corals growing in the Sydney Harbour. All of these blobs that we see on this rocky reef are coral. And although it's exciting and interesting to see this, I also want to remind us of what reefs look like up north. Very, very, very different. And the point that I wanted to make for this comparison is that just because we have gains in some places and losses in other places, and maybe on average, we don't see that much change. The gains and losses are not necessarily the same, and the average probably doesn't tell us that much. So I think what we can take with a great degree of confidence is that things are changing fast in their composition, and they're changing in lots of places and producing different combinations of species, etc. But we should definitely not take from this that that the gains and the losses are necessarily comparable or the same, or they cancel each other out. Okay. We often focus our analysis, like when we look at both the turnover and the species regions that I've shown you so far, are largely focused on colonizations and extinctions, but our data allows us to look beyond that. And the point that I wanted to make is that these correspond to a very small fraction of the populations that we have, of the species that we have in our assemblages, 5% and 3% respectively. And the overwhelming majority of the other species that we see in assemblages, the 92% of the rest, are either things that are always there or that blip in and out, so appear and disappear multiple times within our even our relatively short time series. And moreover, in addition to that, there's a great deal that is happening in these populations, with some of them increasing and some of them decreasing. Again, as is kind of becoming a bit of a norm in papers that I am involved in, these are largely centered on zero, in that we have increasing populations in roughly the same numbers as decreasing populations. And this is true for both the persistent and the multiple extinctions and colonizations species, but less so for the colonizing and the extinction, the ones that are going locally extinct. So given all of this change, both in terms of population sizes as well as the identities of the species that are present in any one place, a question that we're starting to ask at the moment is, what does this mean in terms of the traits that are present in assemblages? And we currently I'm currently co-leading another essay working group called S-Tetra. I'm co-leading with Franzi Schrott. And the next few slides that I want to show present are not published yet, but present an analysis that we have done that is focused specifically on body size. And this is work that is being led by Inês Martins, who's also co-leading the working group, I should have said. So we subsetted the data that we have within BioTIME to focus on time series where we have both biomass and abundance estimates because this allows us to estimate mean body size for each species present in the abundance. It's a good estimate, but it is at least an in situ observation of a trait that we are particularly interested in. And body size is a very important trait for all kinds of reasons, from metabolic reasons to biogeographical reasons. There's many reasons why we think body size is important, and also there are multiple lines of evidence suggesting that it is decreasing through time, including extinctions of megafauna, etc. And indeed, when we look within this data, we see again a great deal of variability in the slopes and our estimates of the slopes of change in body size through time. But we see a higher prevalence of shrinking than increasing body size. But what I find even more interesting is that if we look if we try and dissect this change and look at what part of it is associated to changes of the average body size within species versus changes that are associated with the replacement of species with other ones, we see all kinds of different combinations. Again, there's a higher prevalence of shrinking body sizes. You see much higher density of points in the bottom left triangle of this plot, but we see all types of different combinations. Okay, so talked about what data we've been using. I've described some patterns that we see running out of time, and I would like to end with a couple of reflections. My first reflection is that we see biodiversity change, but it's complex and nuanced. We can see that some places are losing species, some places are gaining species, but there's turnover pretty much everywhere. We can see that there are some winners and losers in this prevailing pervasive biodiversity change. We see some evidence that this change is not random. For example, we see a signal of shrinking in body size, but it's messy. It's complicated to see the patterns, and there is a great deal of noise in the data. Which leads me to my next reflection, which is biodiversity data are both messy and precious, and the two things can be true. And they're messy because they're biased, they're messy because they're incomplete, they're messy because each measurement is an estimate or a measurement, and therefore it's ridden with error. And so there's lots and lots of noise in this data, but there is also signal, and some signals are emerge if we look carefully enough. Having said that, despite the messiness, the data we have, and the data we have from the past in particular, is precious because we can't travel in time. And there is, after all, some signal in these shadows, and we can learn some things about how biodiversity is changing, despite all its messiness. But the data are also precious for one more reason, which leads me to my third reflection. So, having spent the last 2 weeks observing people doing observations, it kind of I became more keenly aware that actually every single one of the millions of records that have gone into these analyses come out of humans observing nature, and that made me reminded me that actually observing the natural world and collecting data is really a wonder and a privilege. It puts us in times and places and allows us to see things that are really quite amazing. This is a photo that I took last week. We had just stopped at our safety stop where we had to spend 3 minutes to try and get rid of some of the nitrogen that had accumulated in our bloods. And out come a pod of dolphins who were observing us as we were observing them. And this is not data, like they were not part of our observations. They will not be mentioned, this dolphin encounter will not be mentioned in any papers that we might write with the data that was collected. But I have no doubts in my mind that probably the most memorable moment of the last couple of weeks, both for me as well as the students who were with us in the field. And with that, I want to end my talk by saying thanks to all the people whose work I hope I haven't misrepresented in this talk, the BioTIME core group, as well as a number of long-term collaborators, and as well as home institutions and funders. Thank you.
Thanks, Maria. I hope you can hear me now. Okay, so Sarah, are you around?
Yes, David.
Okay, maybe can you put again in the screen the slide about